In this video, we will discuss about the pathogenesis of non-alcoholic fatty acid, how the persons or teetotaler persons can develop these fatty acid problems. So it starts with a condition that is nutrient excess condition. So what happens in this nutrient excess conditions that it will trigger the insulin resistance condition in this adipose, and this insulin resistance condition in this adipose will leads to the excess free fatty acid surrounding the in the periphery of the liver, or which are easily accessed by the liver. And usually when the liver is in a insulin resistant condition, what happens is that it will generate de novo lipogenesis process. At the same time, it will inhibit the glycogenesis process. However, in this case, the insulin resistant condition is on the periphery, that is insulin resistant condition is on the adipose. So in this case, the de novo lipogenesis is continues to occur, and at the same time glycogenesis is also continues to occur. And because of that, because of the continuous glycogenesis also formations, there is a increase in the triglyceride formations occurs within the liver, and that will create oxidative stress, and this oxidative stress create additional pressure on endoplasmic reticulum to generate more proteins. And within this endoplasmic reticulum, as we have discussed many times, that there is a quality control mechanism occurs that take care of this the protein folding, and this condition will lead to the additional pressure on the ER to generate more proteins, and that will create more additional stress on the unfolded protein response. Or we can say that the more number of proteins are found, this leads to the more number of unfolded proteins also, and that will create the ER stress within the cells. And because of this, several signaling pathways get triggered, such as pathway IRE1 pathway, and this because of triggering of this pathway, that will lead to the upregulation of the genes that are involved in the inflammations, or pro-inflammatory genes will get upregulated, such as this CHOP protein. Because of the upregulations of this uh, PIKK pathway through this IRE1, which leads to this P65 P50 upregulations, and that will cause the upregulations of this NF kappa beta S, that will lead to this uh, that will lead to the translation or transcription and translation of this CHOP protein, which is a pro-inflammatory genes, and that will cause more excess inflammations within the livers. So this conditions, which starts the inflammation of from this in the periphery because of the adipose, will cause further inflammations within the liver. Uh, so I forgot to tell you that because of the insulin resistance, there is a inflammations will be generated on the adipose. So this will cause the further inflammations that can be generated because of the cascading effect due to the release of free fatty acids, and at the same time the uh, de-inhibition of this glycogenesis. So these things can be taken care of by different different chemical agents, such as this Ml127, Ml351, Ml335. It helps to prevent this uh, prevent the translation of this inflammatory genes that are being marked here, such as this CHOP proteins or the other proteins that are regulated because of the ER stress. And at the same time, it also helps in inhibiting the glycogenesis process. So this is how the teetotaler persons can develop non-alcoholic fatty acid conditions because of the excess nutrient conditions. And this can one of the examples that this can occurs is because when the peoples are doing starvations or fasting beyond the limit by that can be handled by the that can be handled by the person. So, so I hope you got a basic idea about this non-alcoholic fatty acid pathogenesis. And if you need to understand more about or in detail about this pathogenesis process of non-alcoholic fatty acid, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.